Okay. Well, thank you for the introduction, Danny. I have the pleasure of, Nyssa gets two introductions. So, uh, Nyssa is a PhD student at the University of Illinois, Chicago, working in Brian Murphy's lab, and so she's close to graduation. So, as an aside, if you're impressed with the work, you know, and you're hiring. But anyway, no, she's been a great collaborator, and she's got a background in chemistry, and so this has been a really fun interdisciplinary collaborative project that she's been spearheading, called IDBac for on a web platform. So the next generation of IDBac, and she'll be giving you a highlight of what's there, some of the methods, as well as a little walkthrough and all the hard work she's put into this over the past couple of years. So without further ado, Nyssa, floor is yours.
Right, well, thank you for that kind introduction. I'm happy to be here. I'm excited to talk to you all today about the IDBac Open Access MALDI-TOF MS database and analysis web platform. So to start off, we covered a lot of the background here, who we are and how IDBac came to be, but I'll give you a few more little details. As mentioned, my name is Nyssa Krull. I'm a PhD candidate with Brian Murphy's lab at University of Illinois, Chicago. And we are a drug discovery lab that focuses predominantly on bacterial and natural products. To do so, we go into the environments and take samples, whether it be moss and detritus from a forest in Wisconsin, a hyperalkaline pond in a Chicago Park District, or traveling to the freshwater marine environments off of Iceland to all look for the next bacteria that will produce the next drug lead. And one of the strong aims in our lab is to increase efficiency and decrease phylogenetic and chemical redundancy on the front end of the drug discovery pipeline. So as you might imagine, you go out to the environment, you take samples, you come back to a lab, you work up all these samples, you do a lot of plating, and you end up with hundreds to potentially thousands of bacterial isolates that need to be processed for, again, looking for that next drug lead. It became very clear that we need a rapid, reliable, and cost-effective way to identify bacteria from the environment. So what are our options to ID these bacteria? We can go the colony morphology route. We can look at the plates, we can look at color, size, opacity of these colonies. And this has been one of the mainstay and first ways of identifying bacteria in microbiology. You can get some information, but clearly just appearances don't tell the whole story. So we can move on to technological advances such as 16S rRNA gene sequencing, where we have the ability to highlight this highly conserved gene within bacteria. And while this has become one of the most common methods in identifying bacteria, it becomes cost prohibitive when you are working with, again, hundreds to thousands of bacterial strains. And it also just gives you a little glimpse into the bacteria that you're working with. So there's whole genome sequencing. Whole genome sequencing is arguably the best way to identify bacteria. You get the full picture, and for a drug discovery lab like ours, you get a lot of information on that biosynthetic potential. But if 16S rRNA becomes cost prohibitive at a large volume of bacteria, whole genome sequencing is just out of the question for the volume of bacteria we work with. So our lab started focusing on another method, which is MALDI-TOF MS. To give you a little background and a brief history of matrix-assisted laser desorption, ionization, time-of-flight mass spectrometry, or more simply MALDI-TOF MS, the use of mass spec to identify bacteria began in roughly 1975. MALDI MS was developed in 85, with TOF being added in 1988. A little under a decade later is when we started seeing researchers use this powerful tool to identify bacteria. In 1996, when there's two publications that came out right around each other, again, harnessing the power of this rapid way to identify bacteria. From there, a little over a decade later, we start seeing global use in clinical laboratories across the world from 2013 to current day. And this makes sense. This is a quick and efficient way. I've given you here the workflow of isolation plate to data analysis. So we start here with picking a colony from pure isolation plates. Using a sterile toothpick, we take a thin layer of cell mass and apply that to a ground steel target plate. And from there, we overlay 1 microliter of formic acid and 1 microliter of matrix. You acquire the data is digitized within the instrument-based software, and then you get a mass spectrum. Low cost, high throughput, alternative to those methods that I mentioned before, like 16S and whole genome sequencing. And to really dig into that a little bit further, I'd like to do a timeline, process and timeline comparison of 16S versus MALDI-TOF MS. And I can tell you, background in microbiology, I've extracted DNA from hundreds to thousands of bacteria. I feel very strongly that this timeline is best case scenario. So we start out with extracting DNA from bacteria. There are quick and efficient ways to do this, but many groups rely on kits for quality data. It could take around an hour and a half. You then have to amplify that 16S rRNA gene through polymerase chain reaction to run a gel electrophoresis to confirm whether or not you had successful extraction amplification. To extract that band out of the gel or clean up your PCR reaction. Move on to sample preparation. Perhaps you're going to hand it off to a core in your facility or prepare the samples to be shipped for that sequencing. Sequencing itself can take 24 to 48 hours in best case scenario. Once you get those data back, you trim your sequence and assemble if you've used 2 different primers. And then finally, you run blast analysis. This compared with the MALDI-TOF MS timeline, you can see quite a difference here. That whole process I showed you on the previous slide, from plate to the formic acid and matrix overlay, that takes roughly 2 hours to complete. And MALDI-TOF data acquisition, depending on the instrument you're working with, takes roughly an hour as well. And then you have immediate data that you can analyze, knock that out in another hour. There's a stark difference in this timeline. For 16S rRNA gene sequencing, these 2 to 3 days that I walked you through is covering roughly 30 strains, maybe a little more, a little less, depending on your experience with this. Whereas the MALDI-TOF MS process and timeline, in theory, you could run through and collect data on 384 strains within roughly 4 hours. Now you might be wondering, how does this identification work? I'll give you a very simplistic overview here of putative bacterial identification through spectral similarity. The overall gist is protein spectra are collected from roughly 2,000 to 20,000 Daltons, and ionizable proteins within this mass range represent unique fingerprints. The literature has established many of these proteins are ribosomal, so they're highly conserved, though there are other carrier proteins and translational factors that might be ionized as well, are driving these comparisons. Once you have these spectra, you have uncharacterized isolates that are then compared against spectra of genetically verified strains, to which you can then identify similarity or dissimilarity between the two. And I mentioned on the history slide that this process, the efficacy and the rapid approach with MALDI has been adapted by the clinical laboratories across the globe since roughly 2013. And there are 2 main companies that are kind of driving that field. There's bioMiro with the Vitek system and Bruker Biotyper. Both of these companies possess databases of genetically verified reference spectra, and they also have accompanying proprietary software. And this has been a massively impactful addition to the clinical studies. If you have a patient who is ill, time is of the utmost importance, identifying that bacteria and not taking the 2 to 3 days that I just described to you with 16S rRNA sequencing. It's been a wonderful addition to hospital systems. However, for the broader research community, these systems are proprietary, so they lack transparency. The exact metrics used to assign similarity in the scoring systems are held quite close to the chest. Cost prohibitive as well. While hospital systems might have the budget to spend the roughly $40,000 it takes to acquire the database and the software, many research labs don't have that ability. And finally, there is a lack of taxonomic diversity, with the exception of some groups adding additions of environmental bacteria. Most of the spectra that you find within these databases are focused on human pathogens, since this is focused on clinical settings. Makes sense. So in an effort to try to bridge the accessibility gap, if you're a lab that doesn't have access to the Biotyper system, IDBac desktop platform was created by Dr. Chase Clark out of the Murphy Lab. And within this desktop platform, users were able to build personal libraries of protein and small molecule MALDI-MS data, protein dendrograms, organized isolates by user-defined similarity metrics here in this upper photo. In-house seeds then of genetically verified strains could be used to help identify isolates. It would be entirely dependent on whatever isolates, genetically verified strains that you have in your possession in the lab. And then metabolite association network, or MANs, allow for rapid visualization of small molecule overlap across isolates. And this was a great success. There was over 1,300 downloads across 44 countries. Users were very happy to have the opportunity to have a relatively automated way of processing and analyzing their MALDI-TOF MS data. While it was impactful, the desktop platform lacks a public database. Again, it was only connected to in-house seed libraries and was not made available via the web, which diminishes accessibility quite a bit. And the database component is very, very important. The ability to identify bacteria is reliant on how robust and taxonomically diverse your database is. So we see in the literature, there are several groups that have made efforts to create in-house libraries based on environmental strains. You have anywhere from cave bacteria to sponge-associated isolates to extremophiles. And you see this sprinkling of in-house libraries across many different groups. However, there are some limitations with these libraries that prevent them from being far-reaching resources. The majority of these efforts possess fewer than 100 strains. They represent taxonomic or environmental niches. And finally, these groups that have created these libraries have access to either Biotyper or Vitech to be able to analyze their data and curate these libraries. So again, we're getting that paywall up against that proprietary software. So without a centralized repository, these valuable data remain inaccessible to the broader research community. And adding on here, in the literature, there are many, many reviews that call for databases, and that likely being the reason why this hasn't been adopted by the broader microbiology community, that lack of accessibility to reference data. So here we present the IDBac Open Access Database. I'm going to take you on a tour of this database in just a minute here, but I would like to highlight our contributors and then also Michael Strobel, who is a PhD student in the Lung Lab at UC Riverside. All of the visualizations that you see here on out, from the database to user interface, are thanks to his hard work. And he's also on this call and is around to help answer if you have any questions as well. So the database, highlighting some of the contributors that we have. This is meant to be community sourced. So these institutions down here at the bottom of the screen have been a tremendous help in creating the starting population of the database. We have the University of Chicago, the Duchossois Institute, that possess many microbiome strains. We have the Beauty Lab at the University of Wisconsin-Madison that have many strains of the family Micromonosporiaceae. We have a collaboration with the NASA Jet Propulsion Laboratory, who have many samples ranging from clean rooms to pre-flight spacecraft swabs. This has been a really fun and interesting collaboration with the JPL. We have the Metcalf Lab at the University of Illinois, Urbana-Champaign, and in tandem with the USDA that have provided the NRL collection. So several hundred isolates of streptomyces that have been contributed into the database. We have the Livingston Lab at Simon Fraser University in collaboration with the Eustachio Lab at UIC that have contributed Burkholderia and Paraburkholderia species. And we leave a little section here, your picture here. Again, this is a community effort, so we are hoping to have community members that would like to help us out, contribute. If you have any questions or if you are interested, please feel free to send me an email. I think maybe we can add that into the chat. I would love to talk about what isolates you might have. So for this community source database, we wanted to make things easy on the researcher and the contributors. So these database depositions require minimal efforts. You have these three main components. You have protein spectra in a file format that's MZML. And if you need any help in converting from raw data to MZML file format, we do have a documentation page that walks you through the process and gives you resources on how to do so. Then we have here a metadata sheet where we ask for some of the basic name of the strain, GenBank Ascension or 16S sequence, whatever culture collection, whatever the PI's name associated with this strain is. We fill out this sheet, which I think many of us have Excel docs with this information already. We have a nice copy and paste scenario. And then we have the deposition workflow that is hosted on GMPS2. You can see here we have just a few different fields. You add in a job description title. This is for your reference, knowing what the collection of bacteria is. You can input your metadata file, which is this, and then you input your spectra. And that's it. You hit submit, and you are then able to go visit the database. So a little live tour of the database. This is our current landing page. Up top, you can see that there is a table that is highlighting the bacteria that we have in the collection. It is the same information that you saw in that metadata sheet I just had on the previous slide. You can go through and check out what bacteria is currently in our database. You can also, if you are not interested in culture collection, you can untoggle this, or you can add any of the columns that are most interesting to you. If you click on a strain, you can then scroll down and visualize the spectra. Below that, we have additional information that users might find helpful, such as the full taxonomy breakdown. And if there's an ascension number added or sequence that was added, you can find it down here. If we look over to the right now, try to not scroll up and down too many times for you all. Here we have a taxonomic tree that represents all the bacteria that are present in our database. And my favorite is this pie chart that breaks down the genera or species that we have currently in the database. Now, Michael's work has made this so that every time there's strains deposited in the database, these figures will be updated. So it will give you a view of the current status of the database. So I've shown you what's in the database. I've shown you that database page. I want to give you an application of this. I'm gonna stay on the drug discovery angle here. Again, we collect many different bacteria from the environment, and here is a representation of that. I have a protein dendrogram on the right-hand side here of 426 uncharacterized environmental isolates. Now, for drug discovery, there are certain genera that are of interest. Perhaps they have large genomes. Perhaps there have been publications on interesting biosynthetic gene clusters. There are many genera that we would like to target from the environment. And again, trying to use sequencing on this many strains is just impractical and would take quite a bit of time, and is untargeted in general. So searching for Micromonospora, a genus of this family, Micromonosporiaceae, we have a database seed here that was contributed by the Uni Laboratory, University of Wisconsin. And as you can see here, there is clustering of 2 uncharacterized strains with this seed. In this scenario, I am more than happy to extract DNA and run 16S or whole genome sequencing on these strains because it's 2 out of the 400. And that's what we did. We ran 16S RNA sequencing, which did confirm that these both were Micromonospora species. This is showing a way where you can mine specific taxa from a large data set with minimal time and efforts. And moving from there, I'd like to start highlighting the analysis platform where we created this protein dendrogram. So the analysis web platform, as Dan mentioned, and we talked about the desktop application IDBac that Chase Clark created, we have added components that there are very similar analysis options as the desktop application, but we have added a few more that we are excited to show you. Similar to the deposition process, we wanted to keep this as straightforward as possible. We want users to be able to readily and quickly analyze their data. So again, you have MZML data for your protein and or small molecule. You have a metadata sheet where you can even add in any columns that might not be shown here. Perhaps you have endospore bacteria and you want to highlight that on the user interface. Whatever you add here will populate to the user interface, which is a nice feature. And then we have the workflow input, again, hosted on GMPS2. We have a few more buttons here you might notice. This is all for giving users optimization options. So you are doing the same thing. You're inputting your protein or small molecule data. You add your metadata sheet. And if you are incorporating small molecule data in here, we ask for a media control. Then you get down to the parameters that you can choose the distance metric, use cosine, Euclidean, anything from this dropdown. You can look between a specific mass range when you are querying against our database. And you can set the database search thresholds to whatever you prefer. And from here, we are now going to take the live tour. Again, go back to the database, but we're going to look at this landing page and then the analysis platform. So if you want to follow along with me, you're more than welcome. You can go to idbac.org, or you can go save this link for later and explore it on your own time. If we scroll down here, you can have a direct link to the analysis workflows. You can just directly to the deposition workflows to deposit streams into the database. You can visualize the database, that page I showed you just a few moments ago. You can review the documentation that gives you a walkthrough on how to process your data, get it into GMPS2. And then we have the interactive interface. So I'm going again to try to not scroll up and down too much as I walk you through here. So I'm going to keep this zoomed out a little bit. So this is a sample data set. If you are interested in exploring the analysis options that we have available, but you don't have data ready, you can just jump on here and test out all the parameters we have to offer. So up top, you're going to see those parameters that you set in that initial workflow on the GMPS2 workflow. You can scroll down and you have this file warning section, which is quite useful for the user for troubleshooting. This will show you, let's say you have biological replicates of 5. You have 5 different MALDI wells that are collected. If one of those is a flatline spectrum, it will let you know here. You want to ignore that warning, so you don't want them on your page. You can deselect the show warnings, and you can start scrolling down to the dendrogram display options. I'm going to nest these so we have a little better visualization of the dendrogram here. So to start out, dendrogram clustering settings. You, again, can choose whatever hierarchical clustering method that you would prefer. You can color code clusters based on dendrogram cut height. As you see here, if I'm using a vertical cut height in the dendrogram, about 7 here. Anything at or below 0.7 is going to be individually color coded. You can see that change as we drop down that cut height to 0.3. So bring that back up maybe. Then we get into the metadata. If I'm going to highlight back up here, if you did not submit a metadata sheet during the workflow, or perhaps you need to edit your metadata sheet partway through and after the workflow has already been submitted, you do have the option to drag and drop a CSV or an XLSX file here. And we will go back down to the metadata. We have 2 visualization options for the metadata right now. One option is to select a metadata category that will be displayed as text next to our strain. So you can see here we have the PI listed next to the isolate ID. Or we can add in a metadata category that will be plotted alongside the dendrogram. In this situation, I've plotted the isolate source. You can see swab 1, swab 2, swab 3. And I can use this metadata visualization to start to potentially identify trends within my data set. You can also use this for cultivation media, any metadata category that you wish. And I'll get to that database identification that I showed you with those Micromenospora previously. So you might notice here, I have 1 hit, and this is marked as DB results. If you have a database hit within the threshold that you populated on that workflow, it will show up with DB and then whatever metadata category that you prefer. So none, you see that taxonomy disappears. I'm interested in seeing what that seed isolate is, like DB taxonomy, and there you go. If I'm interested in seeing the dendrogram distance, I can click this and see that these 2, my unknown isolate is 0.2 distance from the microbacterium database seed, so I can follow that up however I please. If you wish to remove database entries, you can deselect all, and you also have the opportunity to look for specific database genera. Perhaps I'm only looking for Micromenospora again, or Streptomyces. I can deselect all and then search for those specific genera to see if I have any hits within the database. And you can also manipulate the database distance threshold. If you increase the distance, you might start seeing more database hits populate your dendrogram. So you can find what range is best suited for your data set. And then as we move down here, the general section, I already showed you the dendrogram distances, but you can also have two other options. Let's say you weren't happy with one of your spectra for one of your strains. Instead of going and rerunning the workflow, you just want to look at the data and then perhaps run the workflow again later. You can deselect certain isolates from your dendrogram. You can also add a vertical dendrogram cut height, which again might help you start identifying different relatedness trends throughout your protein dendrogram. And we'll scroll down here. I like this feature a lot. It tells you how many strains are in your dendrogram. And this gets tremendously helpful when you're working with those massive data sets like we do in the hundreds. Okay. I'll pause here with a lot of information on the UI. Is there any questions immediately about this protein dendrogram landing page before I move on to the other options? Okay. So we're going to move down to small molecule analysis options. I showed you on the previous slide about the IDBac desktop application. We had those metabolite association networks for MANs, and you were able to subtract a media control, and you were able to add certain parameters, again, looking for a specific mass range, but those analysis options were relatively limited. So here we have a lot of options. I don't want to click through too many at once on you, but if we just scroll down here to start, we have metabolite association networks. Each of these large nodes are representing an individual strain, and each of the little orange nodes are representing unique M over Zs. If a strain, as you can see with main principles of networking, if one of these M over Zs is not connected to any of the other isolates, whoop, we zoomed out too much, then it is not shared by isolates in that data set. I'm gonna go back. I zoomed out way too much. My apologies. Let's go here. So let's go through some of the analysis options that we have available. You can manipulate the relative intensity threshold. So if you have that relative intensity threshold quite high, you might see fewer strains or nodes within your metabolite association network. If you decrease that, you might see quite a bit more. We'll drop this down to 0.04 and we'll give it a second to run. Up top, this platform has this useful tool where it'll say running at the very top of the page to kind of give you an idea of the current status of the analysis. And while that's running as well, we're going to look at the other metabolite association network options. We have different network layouts. You see we have 4 or 5 here available. You can incorporate spectral similarity into node layout. So if the protein spectra is similar, that can be a part of how the metabolite association network is organized. You can manipulate the different coloring systems on the metabolite association network itself. And you have a long list of colors that you can choose from. We would like to give everyone the opportunity to create figures that can be used in presentations or perhaps in publications. So you can see once I drop that relative intensity down, we see quite a bit more M over Cs. And again, drug discovery angle, I can give you an example here on how I might use this. Let's say all of these isolates, all of these larger blue circles representing strains are highly, highly related strains. They are all of the same species. And instead of working up and doing chemical extractions, bioassay guided fractionation on every single one of these isolates, I can use a metabolite association network to approximate the chemical overlap between these bacteria. You can see all the nodes here, perhaps a couple of these strains, they seem to share a lot of M over Zs with their neighbors. where there are a few isolates that really stand out and have unique M/Zs of their own. These might be strains that I would prioritize for the drug discovery process. Okay. And then we've covered most of the analysis options up here for the metabolite association network options. And this section of the visualized small molecule data is really there for you being able to decide if you want to look at specific clusters in your dendrogram. Say you're only interested in a few strains, or you only want to compare two strains to each other. You have the ability to populate your metabolite association network however you see fit. And then I'm going to scroll back up and continue down this list. There's a few more pages to show you here. This one is pretty straightforward. We have a plot spectra page where you can visualize one of your strains if you would like, or you can create a mirror plot comparing two strains at a time. In this, you will see there are blue lines in the spectrum and green. The green here is indicating match. And one other feature that I also like too is that you can change the title of your figure, and you can export this as a PNG for later use. And the last analysis page that I'm going to cover here today is the protein dendrogram heat map, or protein heat map, the accompanying dendrogram. So similar to the metabolite association page, we wanted to make this customizable to the user. You want to look at 2 strains, you want to look at 50, you have the option to build your heat map however you see fit. You can go by dendrogram cluster. You can, and we have this little reference up here too, if you need a reminder of what clusters and what their naming convention is. You can search by metadata. Perhaps I only want to look at sponge-associated bacteria and their M/Z patterns in a heat map. I can choose that from my isolate source. And then you have the metadata values next to it. So you can, again, select something very specific like the sponge-associated data. Over here, you can create heat map by strain, incorporating 1 to as many as you would like to populate the heat map. You can remove any that you do not wish to see. And as we scroll down, this is where we get into, again, more user custom parameters. You can look for your replicate tolerance mode. If you have replicates of 4, perhaps you want 50% or 75%, you can work with that up here. You can, again, adjust the relative intensity, which is shown in a scale down here, color-coded scale. You can search for specific M/Zs. And again, another one of my favorite aspects of this analysis platform is incorporating the protein dendrogram. This can be used to start analyzing trends within your data set. Like in this situation here, I'm seeing this cluster that is sharing this M/Z. I'm seeing a cluster here sharing this M/Z. I can zoom into that specific mass range if I would like, or I can zoom out and look at the larger picture. And another feature that is quite nice here is that if you would like to export your heat map, you have the ability to download that into an Excel file that will show you all of the M/Zs present, the strain IDs, and the relative intensity of each of the M/Zs. Let's scroll back up. With that, I'm going to move into the acknowledgements and showing you all the full IDBac team. I think there might be some questions on the chat that I need to get to here as well. But first, I want to, again, thank you all for your time in attending this workshop. I want to highlight those contributors to the database that have generously contributed diverse strains into this effort. And we have our team. We have Dr. Ming Wang and Michael Strobel, University of California, Riverside. We have Dr. Laura Sanchez and Robbie Shepard from the University of California, Santa Cruz. We have my advisor, Dr. Brian Murphy. And we have Dr. Chase Clark, who we couldn't have gotten here without him and creating the desktop application. Down here, we have this little exit survey. If you would take the time to complete this, that would be fantastic. If you're interested in IDBac, if you have any more questions, this is a really nice way to get a hold of us. And there's also the ability to be on an email list for any future workshops that we hold. Some workshops are very hands-on. We dig into actually submitting the strains, and we will hopefully have a schedule of those coming up quite soon.
Okay. Well, thank you, Nyssa. Let's give her a round of applause or a virtual round of applause. I will say, I've seen the evolution of this talk from Nyssa, and number one, it's great from just giving a talk, but I thought the slides are way prettier than anything I put together. So I just wanted to throw that out there.
Thank you.
So that was wonderful, and I'm a little biased, so definitely check it out, and we'll take questions at the very end. And this is an ongoing process in terms of building the infrastructure and building the whole ecosystem. So it'll evolve, but Nyssa and Michael have done a fantastic job, and definitely also other collaborators, Robbie and Chase throughout the years. So. And there's other workshops if you would like to attend, and office hours that Nyssa holds on a weekly basis if you want to start interacting with this. And no pressure, she's getting her paper out soon for this stuff too. And we'll take questions after this, but thank you all for being so engaged. There's a lot of questions in the chat. So I think we'll end the recording here, and thank you very much.